we're just going to dot this thing together because the FBI and the police and everybody else who was supposed to do their jobs covered it up and they protected the criminals. So let's go. I know everybody's doing this. Everybody's dissecting these files until we all piece it together and put it all out there, and they can redact as much as they want. We're still going to do it. I brought up Deutsche Bank and I guess Paul Morris. To this point, the technology known as Crispr. I know non-vaxxer talked about CRISPR, I think. Probably a lot of other people too. This email is from 2015. There's that year again. CAS9 has been a science project, a research tool with enormous potential. You know, in 2015, I was injected with something against my will in the middle of the night. I have the scar to show where the injection was. I don't know what was injected into me. And significant questions to answer on which venture capitalists have placed bets by forming a group of startups. The jackpot, CRISPR, CAS9, a method of performing precise genetic surgery, might yield treatments for a wide array of previously intractable diseases. We're still a long way from anybody claiming that prize, though. No CRISPR therapy has ever been tested in a human being, and a whole lot could go wrong when that happens. Emerging technologies, after all, go through their ups and downs, but today, some of the biggest names on Wall Street and elsewhere are showing that they like the odds by handing the lowest round of funding yet to a CRISPR startup. Largest. Excuse me, not lowest, largest. Cambridge-based Editas Medicine is announcing a $120 million Series B round led by Bill Gates' chief advisor for science and technology, Boris Nikolic. The list of financiers teaming with Nikolic reads like a Rolodex of so-called crossover investors who invest in both public and private entities and corporate venture arms. Among them, Deerfield Management, Viking Global, Fidelity, T. Rowe Price, Google, Jennison Associates, Khosla, ECOR1 Capital, Caston Capital, Omega Funds, Cowen Private Investments, and Alexandria Ventures. You know, Rob McGuckin lives in Alexandria. Editas founding VC backers, Flagship Ventures, Polaris Partners, and Third Rock Ventures also pitched in, as did Partners Innovation Fund. Nikolic, who is joining Editas board, made the investment through what's been called BNG0 or O, a new US-based investment company backed by large family offices with global presence and long-term investment horizon, and formed specifically to invest in Editas. CEO Katrine Bosley confirmed that Gates is one of the individuals investing in Editas. It sounds like we should be looking into all of these names. Oh, I see some RNA coming up. To be clear, while this is a significant round and is not even close to the largest financing round for a biotech startup, during the latest boom, we've seen messenger RNA drug developer Moderna haul in a record $450 million. And the now public cancer immunotherapy company Juno Therapeutics, NASDAQ Juno, which Editas recently partnered with, got $310 million last year before taking itself public. But the round is still the largest financial investment made yet in CRISPR startup, adding to the quickly gathering momentum of the field's fledgling companies. In Intellia Therapeutics and Crispr have made strides as well. Intellia raised 15 million and then cut a broad collaboration with Novartis, another big name. Crispr hauled in 64 million in April in a round that was led by Celgene and the venture arm of GlaxoSmithKline. So this search I did was emerging technologies in the Epstein files, and we're going to do a search on DNA. We know that trafficking is a component, but obviously just trafficking kids doesn't shape the way that you think or control the way you spend your money or control where you go to school or manipulate your whole life, right? It's broader. It's a much broader system in place that obviously has to do with emerging technologies and biotech. Edidas has become the first of the group not only to attract crossover backers, but to begin discussing the diseases that it's targeting. Its first program, Bosley says, is a potential treatment for a form of Leber, congenital amaurosis, genetically driven blindness. It is a different form of the LCA than gene therapy company Spark Therapeutics is targeting. Bosley says the one Editas is going over can't be solved by gene therapy. Beyond that, Editas ongoing immuno-oncology work with Juno. Editas has done some very early work in Duchenne, sorry about all the pronunciations, muscular dystrophy, and exploring ways to repair a mutant hemoglobin gene, something that could have an impact in a range of bleeding disorders. That doesn't mean this is where Editas will ultimately focus. Bosley notes, for instance, there are big technical challenges of producing an effective CRISPR therapy, but it's a glimpse into the company's thinking. Bosley won't estimate how long it'll be before the first Editas therapy begins human testing, noting that the company is in the midst of preclinical work testing its technology in patient cells. We have a little bit more work to do before we can really be explicit about a specific timeline. Does that mean a few years? I don't think it'll take that long. We'll move sooner than that if we have a construct that's good enough. For those new to the story, CRISPR is a two-part system derived from a defense mechanism that bacteria use to fend off viruses. Think of it as a pair of molecular scissors, an enzyme called CRISPR-associated protein 9 or Cas9 being carried into a cell's nucleus by a strand of RNA that serves as a guide, clustered, regularly interspaced short palindromic repeats, aka CRISPR. Once there, the scissors may be able to snip out a defective gene and perhaps replace it with a new functioning one. So, what they're talking about, I believe you can look up, in fact, is similar to, if not exact, the mRNA graphene oxide cloud seeding that's going on. And I believe that's all connected to the smart dust nanotech. And again, go look it up and see how similar this definition is to what they're alleging is cloud seeding. And don't just look up cloud seeding. Make sure you put the graphene oxide and mRNA in there too. Let's see. Bosley says the company aims to make 2 specific cuts in 2 different DNA sites to eliminate the genetic mutation causing the disease. And so I think this is getting into what maybe the business connection, quote unquote scientific research that the broader picture of what's behind the Epstein files. This isn't the first gene editing technique to emerge, and its zinc finger nucleus platform have the most advanced gene editing candidate, a potential therapy for HIV in phase 2 testing. But CRISPR has taken the medical world by storm because of how easy it is to use and the broad potential it may have. CRISPR technology has already been used to modify the genomes of plants and animals, but that ease of use has also led to some serious ethical questions. You don't say. One of the field's pioneers, UC Berkeley's Jennifer Doudna, do not do this, and several others have called for a moratorium on using CRISPR to edit the human germline. Did they just call us germs? Making changes to sperm, eggs, and embryos that would then be passed along to future generations and have warned against altering humans for non-medical reasons. So, guys, I mean, what is the global standardization of biodigital convergence? Human augmentation. Just, they've been doing it. There are some practical concerns when it comes to using CRISPR for therapeutics. How can a therapy using this non-consensual, by the way, they just did all this without asking anyone, with our money. Using this technology can be delivered into the body effectively. How can a therapy using this technology be delivered into the body effectively? Will it safely do its work or cut DNA in the wrong places? One wrong SNP, a so-called off-target effect, could cause serious unintended consequences. Like what? Like a heart attack? And ensuring that this doesn't happen is just one of the technical challenges that have to be faced before CRISPR drug begins human testing. One need only look at the up and down history of gene therapy and RNA interference drugs to see the roller coaster likely ahead as researchers try to figure out how to use CRISPR for therapeutics. While Bosley acknowledges the challenges to come, she notes that part of the excitement surrounding CRISPR is that it's come at a time when our knowledge of the genome is just at a fundamentally different place than it was many years ago. She adds that a lot of progress has been made to combat potential off-target effects, like figuring out the exact right size of the RNA guides and which types of enzymes to use. Meanwhile, Editas co-founder Keith Jung has developed a tool called GuideSec to track instances of unintended DNA cuts. And as for delivering these treatments, Editas isn't trying to reinvent the wheel, Bosley says. Rather, it's looking to proven delivery methods, at least initially for the LCA program. It's delivering a CRISPR Cas9 using adeno-associated virus, a delivery factor that has been used by a number of gene therapy companies. It could use other established delivery technologies like lipid nanoparticles, often used to shepherd RNA interference drugs into the body, or electroporation, in which an electric pulse creates tiny holes in cells that allow drugs to gain entry. Yeah, and I would say that's absolutely what's been going on. I mean, Trump in his second term, first week, said, cures for cancer and all billions of dollars to Oracle. And so this just kind of sounds like what they're doing with that kind of technology. And what's it called when you gene edit the population? There's like a word for that. There's definitely a word for that. I'm just drawing a blank. Still, delivery is a critical challenge in this field. There's no question about that. Who's looking at Lockheed Martin? How are those energy weapons doing? What's going on with the cloud seeding? Are those chemtrails? A patent battle between Editas and Doudna's group at UC Berkeley is also part of the mix. The U.S. Patent and Trademark Office awarded the first CRISPR-related patent in 2014 to the Broad Institute of MIT and Harvard. Hey, Larry Summers. For work led by the boards. That says Broads. That says Broad. Well, led by the Broad's Feng Zhang, an Editas co-founder, the Berkeley group is fighting the patent, claiming it made the invention first. Doudna's work is licensed to Caribou, which in turn has licensed use of its technology for human therapies to Intellia. The work of Doudna's co-inventor Emmanuel Charpentier is licensed to CRISPR, and Doudna herself was an Editas co-founder, but as MIT Technology Review first reported, later cut ties with the company. When asked about the patent case, Bosley didn't give an update directly, but said that the company has a broad portfolio of IP that it's licensed in, and that it's developing patent applications from its own internal work as well. All of which is why the progress of Editas and its rivals will be so closely watched. And why the financing today market such as noteworthy step for the technology. Crossover bracket backers have been increasingly active during the biotech boom, joining up with early stage companies to lay the foundation for a number of public offerings. Editas has become the first of CRISPR Cas9 group to amass that kind of support, but deciding when to take the leap to the public markets is critical, particularly for a company with a new and unproven technology. Moderna executives, for instance, contended that they were not thinking of an IPO in the short term when they raised $450 million. Bosley also brushed off thoughts of an IPO, at least in the short term, while Editas almost certainly have to tap Wall Street at some point to build the broad type of company it hopes to be. There's much work to be done first. That means adding a significant number to its roughly 40-person staff, refining its strategy, and using some of the $120 million to bring several programs to clinical testing. We are on a marathon here at Editas. As much as we think there's some near-term possibilities of things we might be able to address in a more straightforward way, there's a lot to do to really develop the platform. And this is from Paul Morris, again at Deutsche Bank. And sorry for the long freaking video to read that. Just focusing on the technology and connections that are involved in the recent release of the files. Big names, big institutions, and CRISPR technology. Guess that's gene editing.